Section 6. Genomics and proteomics have transformed biological inquiry and applications. The structure of the DNA molecule was described in 1952, and the linear sequence of nucleotide bases was understood to specific the amino acid sequence of proteins. The first chemical techniques for DNA sequencing were developed in the 1970s. Between 1990 and the early 2000s, the entire sequence of the full complement of the human genome was sequenced. We call that the Human Genome Project. Rapid development of faster and less expensive sequencing methods have led to the sequencing of numerous genomes other than humans. A new discipline called bioinformatics was developed using computer software and computational tools. It was formed to handle and analyze the large quantities of data that were generated from these endeavors. Bioinformatics uses computer software and other computational tools. Genomics is the approach of addressing problems by analyzing large sets of genes or comparing whole genomes of different species. Similar to genomics, proteomics is the approach that analyzes large sets of protein sequences. Making connections, contributions of genomics and proteomics to biology include paleontology, studying evolution, studying species interactions, conservation biology, and medical science. Paleontology, we can see where did people come from, where did human beings come from, how are we related to Neanderthals and the Homo erectus. Evolving the hippopotamus from the short-finned pilot whale, how are those two evolved from one another? Making connections in medical science. Conservation biology, how can we use genomics and proteomics to help conserve the species that we still have? And finally, species interactions, how are they related to each other? We can use DNA and proteins as tape measures of evolution. Genes, or DNA, and their products, proteins, document the hereditary background of an organism. Because DNA molecules are passed from parents to offspring, siblings have greater similarity in their DNA and protein than do unrelated individuals of the same species. This argument can be extended to develop a molecular genealogy to relationships between species. Two species that appear to be closely related based on fossil and molecular evidence should also be more similar in DNA and protein sequence than are more distantly related species. Scientists can compare the sequence of 146 amino acids in the polypeptide chain of the human hemoglobin to sequences in 5 other vertebrates. Humans and gorillas differ in just 1 amino acid, while humans and frogs differ in 67 amino acids. Despite these differences, all the species have functional hemoglobin. From that, we can determine that we are probably closer in relation, genetically and molecularly speaking, to gorillas than we are frogs. Here we can see the comparison between the monkey, the human, and the gibbon. This is the alignment of the amino acid sequences of the beta globin. Each of those letters represents different amino acids. The Rh factor in your blood type refers to a surface antigen that's similar to the one on the blood cells of a rhesus monkey, and they were used to study the interactions within blood types during transfusions. It's a complicated system, but luckily we only concern ourselves with the D antigen, so we refer to people as being A positive or A negative, with that positive or negative representing that Rh factor. There's actually multiple different ways of classifying blood types, but the AdvBio system using the Rh factor is the most common. You can find the following slides in your book in a table. They're also on the back of your study guide. They can help you organize your information, or you can use the chart that we completed in class. There is one posted in